PharmapPal.
You're listening to Bloomscast, the official podcast of Blooms.io, home of the bioactive library for mass spectrometry instant optimization. Let's dive into the latest in natural chemistry, bioactive discovery, and the future of lab innovation.
Today, we're tackling a really interesting area in skin research. We're looking at thymoquinone, TQ for short. It's a compound from black cumin seeds, and specifically how it might potentially, shake things up for treating psoriasis.
Yeah, psoriasis, it's a condition many people following this field know well, a chronic inflammatory skin issue. It's a pretty complex immune system, genetics, environment, all playing a part. And it leads to those characteristic scales, the plaques, often itchy, sometimes painful.
And it's quite widespread.
Oh yeah, globally maybe 2-3% of people have it. Though it varies, more common in some populations, less in others. Fundamentally, it's immune-mediated inflammation. And interestingly, there's no single diagnostic test. It's usually clinical observation.
And it's not just skin deep, right? There's the arthritis link too.
Exactly. Psoriatic arthritis. It affects a fair chunk of people with plaque psoriasis, maybe somewhere between, say, 7% and nearly 50% in some studies. And psoriasis itself isn't just one thing. There are different types. Plaque is commonest, but also guttate, inverse, pustular, and so on.
Okay, so a pretty significant challenge. Psoriasis. And our focus is thymoquinone, TQ. It comes from Nigella sativa seeds. As a phytochemist, what makes TQ stand out to you?
Well, looking at TQ from a phytochemical angle, its structure immediately hints at potential activity. It was isolated back in 1960, this sort of yellow compound. And one key thing noted early on was its solubility. Poor in water, better in things like ethanol. And that basic property, has huge implications for how it behaves biologically and how we might use it therapeutically.
Right, which leads us to its supposed benefits: anti-inflammatory, antioxidant, immunomodulatory. How do these link to psoriasis?
Precisely. Psoriasis involves, too much inflammation, an overactive immune response in the skin, and oxidative stress is a factor too. TQ seems to hit several of these points. Preclinical work keeps showing it can dampen down these key processes, like a natural modulator.
But as you mentioned, that solubility issue, if it doesn't dissolve well in water, that's a problem for getting it delivered effectively.
Exactly. Poor water solubility often means poor bioavailability. It's hard for the body to absorb and use it efficiently, especially for a skin condition where you might want a topical treatment. And that's really where nanotechnology comes into the picture.
Ah, the Nano-TQ formulations.
Yes, these tiny, sophisticated delivery systems, basically designed to package up the TQ. Researchers are looking at different types: nano-emulsions, which are like superfine mixtures, lipid vesicles, sort of fat-based bubbles, nanostructured lipid carriers, or NLCs, which are a bit more complex, and ethosomes. Ethosomes are interesting. They use ethanol in the structure to help them penetrate the skin better.
Okay, so the core mission of this deep dive is really exploring how these nano-TQ formulations could potentially transform topical psoriasis treatment. We're talking better delivery, getting it through the skin, maybe sustained release so it works longer, and maybe even targeting specific inflamed areas, all based on preclinical work for now, of course.
Absolutely. That's the goal.
Yeah.
Now let's dig a bit deeper into TQ itself, specifically from that phytochemical perspective regarding psoriasis. Its anti-inflammatory actions are quite diverse. On a molecular level, it seems to suppress several key pro-inflammatory signals molecules like IL-2, IL-4, IL-6, IL-12. Well, interestingly, it can promote IFN gamma, which sometimes has more of a regulatory role.
So it's not just blocking everything, it's more nuanced.
It seems that way.
Yep.
It also inhibits NF-κB activation. That's a major control switch for inflammation, and reduces TOX2 production, which makes inflammatory prostaglandins. Something I find quite interesting, phytochemically speaking, is its effect on things like human beta-defensin 3, other antimicrobial peptides, and LL37. These are crucial for the skin barrier, which is often weak in psoriasis, making it prone to infection.
Wow, so it seems to be working on multiple fronts, the inflammation drivers, and maybe helping repair the skin barrier too. What about the antioxidant side?
Also very important. Psoriasis involves oxidative stress, damage from unstable molecules called free radicals. TQ is good at scavenging these free radicals. It reduces lipid peroxidation that's damage to cell membranes. And crucially, it seems to boost our own natural antioxidant defenses. Enzymes like glutathione peroxidase, SOD, catalase, helps maintain levels of glutathione, the key antioxidant.
So it cleans up the mess and strengthens the defenses?
Kind of, yeah. And interestingly, a related molecule, thymohydroquinone, might be even better at scavenging radicals in some lab tests. This antioxidant activity is linked to something called the NRF2-ARE pathway. TQ can basically switch on this pathway. NRF2 moves into the cell nucleus and activates genes that make protective antioxidant proteins, so it's enhancing the body's own system.
Okay, so multiple mechanisms against inflammation and oxidative damage, and you mentioned immunomodulatory effects too, impacting the immune system directly.
Yes, that's critical for an autoimmune condition like psoriasis. TQ appears to influence both arms of adaptive immunity cellular T-cell responses and humoral antibody responses. If you could see it working, you'd see it adjusting levels of interferons, interleukins, TNF-alpha key immune messengers, often out of whack in psoriasis. It also impacts oxidative stress signaling, influences regulatory T-cells, the ones that help calm the immune system down, and interacts with major signaling pathways like NF-κB, JAK-STAT, and AMPK. From a phytochemical standpoint, its ability to interact with so many of these dysregulated pathways is really what makes it so promising for conditions like psoriasis.
Right. To really appreciate TQ's potential, maybe we should briefly touch on current psoriasis treatments and, well, where they sometimes fall short.
Good point. Current treatments aim to manage symptoms, control the disease, but they all have downsides. Topicals like creams and ointments are often the first step for milder cases, but getting them through that thick psoriatic skin can be tough. Limited penetration. Then you have conventional systemic drugs, like methotrexate. Can be effective for more severe psoriasis, but you worry about side effects throughout the body.
And the biologics, they target specific immune pathways, don't they?
They do, and they've been a huge step forward, especially for moderate to severe cases, but they're expensive. They suppress the immune system, which can increase infection risk. They aren't a cure, and we are seeing some patients who stop responding well over time. Oh, and there's also newer topicals like Tepinarov, targeting the aryl hydrocarbon receptor involved in skin barrier and inflammation. So options exist, but there's definitely room for improvement, safer, more targeted, maybe more sustainable options.
Which loops us back perfectly to nanotechnology. How exactly does putting TQ in nanoparticles address those limitations we just talked about?
Well, it tackles several things at once. For topicals, the big challenge is getting through the outer skin layer, the stratum corneum, especially when it's thickened in psoriasis. Nanoparticles can be designed to enhance penetration. They're tiny enough to potentially slip through or interact with the skin lipids to improve delivery. They can also be engineered for controlled or sustained release. So the TQ is released slowly, over time, right there in the skin where you need it. This could mean applying it less often.
And potentially fewer side effects because it's targeted locally.
That's a major potential advantage. By keeping the drug concentrated in the skin and minimizing how much gets into the bloodstream, you can reduce the risk of systemic side effects you might get with pills or injections. From a delivery science view, it's about getting the concentration right locally while keeping systemic exposure low.
You mentioned different types of nanocarriers earlier. Can we break those down a bit more, like the lipid-based ones?
Sure. Lipid-based ones like liposomes, solid lipid nanoparticles, SLNs, and nanostructured lipid carriers, NLCs, are really popular. Because they're made of fats, basically, they're quite good at carrying fat-soluble drugs like TQ, but can often accommodate water-soluble ones too. They tend to be biocompatible and can improve stability and skin interaction. There are studies, for instance, showing methotrexate in SLNs work better in psoriasis models than standard formulations. Then you have niasomes, similar idea to liposomes, but made from different building blocks, non-ionic surfactants. They can be very stable, maybe cheaper to make. Again, preclinical work shows promise for skin penetration and anti-inflammatory effects with other psoriasis drugs.
Okay, and what about non-lipid options? You mentioned polymeric ones.
Right, polymeric nanoparticles, often made from polymers like PLGA, which is biodegradable and biocompatible. A key advantage here is you can often get really good control over the drug release rate, slow and steady. Studies using PLGA nanoparticles with drugs like tacrolimus or vitamin D analogs have shown better skin penetration, sustained release, and good efficacy in animal models, sometimes with less systemic absorption.
And the ethosomes you mentioned earlier, with the ethanol.
Yes, ethosomes. They're lipid vesicles but contain a high amount of ethanol. The idea is the ethanol temporarily disrupts the skin barrier lipids, allowing the vesicle and its drug cargo like TQ to get deeper into the skin. One study specifically with TQ-loaded ethosomes showed they stayed in the skin longer and had better anti-psoriatic effects compared to just applying plain TQ solution.
It's amazing the different strategies being tried, nanomulsions too.
Yeah, nanomulsions and nanomoldules, the gel form. These are stable, very fine oil and water or water and oil dispersions. They offer good potential for skin penetration and depositing the drug. There's even research combining TQ with fulvic acid from peat in a nanomodule, looking for synergy. And another TQ nanomodule study actually showed it helped wound healing as effectively as a standard treatment, which kind of highlights the potential beyond just psoriasis.
Okay, so a whole toolkit of nanodelivery systems. What's the actual evidence that these nano-TQ formulations work in preclinical psoriasis models?
The evidence is definitely building up and it looks quite promising. Multiple studies in mouse models of psoriasis show that applying nano-TQ formulations can significantly reduce the main signs, the redness, the scaling, the skin thickening. They also consistently show lower levels of key inflammatory cytokines like TNF-alpha and IL-17 in the treated skin lesions.
Can you give an example?
Sure, one study used TQ loaded into dermal lipid nanoparticles. They reported significant reductions in redness, swelling, and skin thickness in their psoriasis model. Another really interesting approach is co-delivery.
Delivering TQ along with another drug.
Exactly. One group developed nanostructured lipid carriers, NLCs, loaded with both tacrolimus and immunosuppressant commonly used in dermatology and PQ. They put this into a nanogel base. They found this combination, nanogel got through the skin better, was more effective against psoriasis signs in their model, and even showed increased toxicity against specific skin cell lines compared to a simpler gel. They used confocal microscopy to actually visualize the nanogel penetrating deeper into the skin layers.
That sounds very efficient, tackling it from two angles with one application.
It's a clever strategy. And it's not just isolated studies. A meta-analysis, which pools data from multiple trials, looked at Nigella sativa products generally. It found they significantly helped with skin disease symptoms, including psoriasis. Other specific studies, like one in BALBC mice, confirmed that nano-formulated TQ could treat psoriasis effectively in that model, with minimal side effects noted. And even in vitro and lab dishes, TQ lipospheres reduced inflammatory markers. This was backed up by an in vivo animal study showing improved skin and lower IL-17 and TNF-alpha, with visible improvement pretty quickly, like within a week.
Okay, the preclinical data certainly seems encouraging, but that's lab work and animal models. What needs to happen next to see if this could actually help people with psoriasis?
Right, that's the critical translation step. We need a lot more research before this becomes a standard treatment. First, comprehensive pharmacokinetic studies how the body absorbs, distributes, metabolizes, and gets rid of these nano-TQ formulations. Where does it actually go? That's biodistribution. And of course, thorough toxicity testing is absolutely essential. We need to be sure these nanoparticles and the TQ they carry are safe for long-term use on the skin.
Dose finding too, I imagine.
Definitely. Dose-response studies to find the sweet spot, the most effective dose with the fewest side effects. And validating the findings in different psoriasis models, like the widely used imiquimod-induced model in mice, helps build confidence. But the most crucial step, really, is well-designed clinical trials in humans. We need rigorous studies in people with different levels of psoriasis severity.
Testing safety, how well people tolerate it, and of course if it actually works.
Exactly. Both as a potential standalone treatment, a monotherapy, and maybe used alongside existing treatments. That combination approach is important to explore.
Looking even further out, are there other research directions? You mentioned combination therapy.
Yeah, exploring nano-TQ as an add-on to current systemic or biologic therapies is a big one. Could it boost their effectiveness? Could it allow lower doses of those stronger drugs, maybe reducing their side effects? That's a key question.
Yeah.
And from the formulation side, the phytochemistry delivery aspect, there's always more optimization to do. Can we design the nanocarriers even better? Enhance skin penetration further, fine-tune the release profile, maybe even add targeting molecules to direct the nanoparticle specifically to troublesome immune cells in the skin, like Th17 or Th1 cells.
Like guided missiles for the TQ?
Sort of, yeah. A bit futuristic still for topical delivery, but it's an area of research.
Yeah.
Also exploring other types of nanocarriers, different lipid compositions, microemulsions, advanced liposomes, and refining that idea of putting multiple drugs like TQ and something else synergistic into one single nanoparticle formulation.
So pulling it all together from this deep dive, it really feels like applying nanotechnology to thymoquinone is, well, a genuinely exciting path forward for topical psoriasis treatment.
I think that's fair to say. By using these nanotechnologies, we have a real chance to overcome TQ's natural limitations, that poor solubility and bioavailability.
Yeah.
And potentially unlock its therapeutic power for psoriasis, leveraging its anti-inflammatory, antioxidant, and immunomodulatory properties, all supported by that growing body of preclinical work rooted in its phytochemistry.
We do need to stress, though, that it's still relatively early days in terms of clinical proof.
Absolutely. The journey from promising lab data to a widely available proven treatment is long and requires really rigorous testing in people. We're not there yet, but the potential is clear.
Definitely. And maybe a final thought for you, our listener, to ponder. Given everything we've heard about thymoquinone's complex actions at the molecular level hitting inflammation, oxidation, immunity, could future research, combining phytochemistry and nanotechnology, lead to even smarter, more personalized therapies? Could we eventually move beyond just managing symptoms of conditions like psoriasis towards solutions that are more, sustainable using these advanced nature-derived approaches?
It's a fascinating prospect, isn't it? Really highlights the synergy possible between understanding natural compounds and developing sophisticated ways to deliver them.
Thanks for tuning in to Bloom's Cast, brought to you by blooms.io. Stay curious, stay connected, and we'll see you next time on the frontier of natural product innovation.
PharmapPal.